infuse oxygen into that tissue. But the rate limiting part of all this is the diffusion. And that's the part that we absolutely have to have the appropriate regulatory system to get the oxygen where it's needed. In terms of diffusion, I teach in my class to undergraduate students that if you have a layer thickness that's 1 millimeter thick, if you change the oxygen levels at the surface and it's sitting on a glass, so it's on glass and you've got a layer that's 1 millimeter, it will take about 10 minutes for that to equilibrate. And it goes as the square of the thickness. So if you doubled it, it'll take 4 times as long to equilibrate. But it also works the other way. If you're going from 1 millimeter down to 0.1 millimeters, a factor of 10, it's 100 times faster. If we go down to 10 microns, whatever you do the math, I can't think up here anymore. The diffusion on these scales, it can be very rapid. When we change our oxygen in our chamber, the saturation of the red cells changes very, very quickly. It's the signaling that takes a while for all of this to occur, and then it's the transport of the red cells that have to follow it to come down to where we've sent this, where it's needed.
Just one more comment. In this whole scenario, I think the sequence of events generally starts up that you get acid rich, and then you go to do it, the mitochondria use it up, and then they want more oxygen. And then the signal comes and goes to now the concentration where you see where the walks and freezes. But if you have a longer period of time, then maybe you open up, maybe by collateral or simultaneous control mechanisms that you open up capillaries and have all that stuff. But I think that it starts always with the need for oxygen. And I don't know how well you control the need for oxygen in your model.
Well, I think. Yeah. Yeah. Okay, let me see if I can summarize this question. Now I have to remember what the question was. How well do we regulate the need for oxygen in this tissue? Because an increase in oxygen normally is occurring, increase in oxygen flow into the muscle is when we have an increase in demand. So if I suddenly start to exercise, Joe walks me home, or walks me here, we start to exercise to get here, then my oxygen demand in my muscle goes up, and so I increase flow. So how well am I regulating my metabolism in my tissue? I think the metabolism in the tissue, the animal is anesthetized, it's on the stage of the microscope, temperature is controlled, all of its environmental stuff is controlled, and we're only changing oxygen. So I'm only looking at one particular control mechanism. If I twitched that muscle, I certainly would get a massive increase in blood flow. And that increase in blood flow, I doubt if it's entirely just due to the oxygen demand. I think there's other factors that play a role when I turn on metabolism that increases flow. And some of those, actually, arterioles will pop open very quickly to deliver lots of blood into that area. I think this mechanism we're studying is how do you make sure the distribution is correct? If I suddenly open the floodgates, I want to make sure that I distribute the oxygen where it's actually needed. And by increasing the blood flow, I get more red cells in there, more signal sources to send signals and say, this is where I need to distribute it. If I just simply, without thought, increase and dilate everywhere, then I'm just relying on what the geometry of that vascular bed is in terms of what the distribution would be. So that would be sort of like having an electric circuit, right? I'm not really worrying about what the level of current flow is in the different parts of that circuit. I need to have something that's going to regulate that distribution. If I suddenly increase the, I don't know if the analogy is correct, I increase the voltage so I get more current going through, but I want to make sure that I distribute it correctly. That's right.
So you have to have to change to get the distribution change, the resistance in the circuit. It's not just all of it. You can't get change most of the resistance.
And that's exactly what the arterioles are doing. They're adaptively changing the resistance within that network. And where does that signal come from? I'm saying the signal has to come from the capillary bed. I think it's the red cell, but it could be a lot of people think it's the red cell now. Not everybody agrees that it's ATP release. There's other mechanisms out there as well. But a lot of people have started to realize that the red cell makes the most sense in terms of oxygen transport. But it's not the only mechanism that's going to be there. We're not just, in muscle, we're not just regulating oxygen movement. We're also regulating temperature in the muscle. We're going to be regulating based on glucose, in terms of the glucose needs in the tissue. There's going to be also, there's a number of other regulatory mechanisms, and I haven't even begun to talk about those, but I'm just focusing on one particular thing. Yes?
Maybe a naive question. If there is a drop in the arterial oxygen,
Yeah.
will that translate the same ratio to the capillary unit? And if yes, will it be quite
Can you repeat the
different?
question, please?
Sure. I'm asking if there is a drop in the oxygen level at the arterial level, will that translate with the same ratio to the capillary unit?
So if I drop the animal's inspired oxygen level,
Yes.
and I took it from 95% saturation
To 85%.
down to 85%, let's say from 95 down to 85%, would I see the same drop in oxygen saturation in my capillary bed? If it's a vascular bed that doesn't regulate oxygen delivery, it will.
Just assume that it does, everything is working fine and the regulatory mechanism is okay.
It should dampen the drop in oxygen level. I don't know, it
The
won't go
same
down.
ratio?
I don't know if it'll be exactly the same ratio. I have, and why?
Assuming, I don't know why I'm asking, assuming that it is the same ratio or whatever it is, do you think that it would be the same from person to person or hugely variable from one person to another?
It would be different. Well, do you know about pulse oximetry? Okay? Pulse oximetry, you know, where they put the,
That's why I'm asking.
Okay.
Coming from that.
so when they put a pulse oximeter on the finger or on the ear, when you change inspired oxygen levels on a young, healthy adult male, probably in his 20s, early 20s, as you drop their FiO2 and you drop their arterial saturation, you have a proportional drop in the signal from your finger or from your earlobe. And they can calibrate that well enough that you can come back and say that it's correctly within, where it's 1% or even less than 1% saturation. And that's why they can use it in patients in hospitals. So in the finger and the ear, in perhaps one area in the forehead, pulse oximetry works beautifully and will track what happens in terms of the arterial level of saturation. If it goes up or down, it will track it beautifully. Those are about the only three tissues that it works on. Maybe others, I don't know about inside the mouth, but I know that when talking to pulse oximetry companies, it only works when they use it on the finger, and on the ear, and on one place in the forehead. And I take it that those are tissues that are not actively regulating their oxygen delivery, because if they were, they would attempt to offset the drop in oxygen saturation. So if I looked at it in skeletal muscle, which regulates oxygen delivery, or I looked at the brain, which regulates oxygen delivery, I don't think the same thing would occur. So that's something maybe we need to talk about later. But the pulse oximetry works beautifully on these particular tissues. They've tried to use it on different tissues, and it doesn't.
When you're talking about muscles, you don't only use skeletal muscles, but they act the same way, they have the same regulatory mechanism as a smooth muscles do, correct?
Well, smooth muscle, yeah, the smooth muscle, depending on what smooth muscle you're talking about, will be usually.
I'm coming from the airway.
Yeah, so I don't, the airway, yeah, smooth muscle in the airway, that's a fun one to work with, because now you've got lots of oxygen around.
The reason I'm asking, I thought that maybe some part of the question, solution, answers come from looking from there is a significant drop in arteriole level, there are people who get out of the apnea by themselves, even if they have, let's say, 200 episodes of apnea. They get out of it by increasing their chemical drive, metabolism, without waking up. So we have people with severe apnea, but without high number of arousals. So they don't have daytime symptom. On the other hand, there are people who are mildly apnea, but many, many times during the night they wake up in order to dilate the airway muscle. So I was wondering, is this drop in the arterial level, how does it translate to the capillary unit? So, because it is the capillary unit that orders the, regulates the airway dilation, isn't it? I mean, they are related.
You're getting me into unknown waters in terms of what's happening in the airways. I'm not as sure of what's going on in the airways in terms of regulation.
Trying to make me interested to make an animal model of the sleep apnea. That's the bottom.
Well, we'll talk about that. Let me just, there's one other related to the lungs at least. And how many have heard of hypoxic vasoconstriction? Okay? What I'm talking about here is hypoxic vasodilation, right, in the periphery. But in the lungs, if you had a region of the lung which is hypoxic because it's not properly ventilated, you don't want to dilate the blood vessels to send more blood there because it's not getting any oxygen. You would make the situation worse. So you want to have hypoxic vasoconstriction to keep the blood away from that area that's not properly ventilated. There's a very recent paper out that talks about the mechanism associated with that. And guess what? It's capillaries at the alveoli signaling to the arterioles to vasoconstrict, to divert the blood flow away from it. And it's actually a conductive signaling within the capillary tree, just like I'm saying here, but with different receptors and a different mechanism associated with it. But once again, it's the capillaries that are signaling the arterioles and changing the flow. What's going on in the airway? It's a very, I don't want to just open my mouth without thinking carefully. So I have to think this one through. Okay, but I will. Any more questions, or are you going to, everybody's kind of tired? We've had some really good questions, and it's a good challenge to respond. It's just like having a PhD defense. Well, thank you very much. Thank you for being very attentive.